First of all, thank you to the organisers for the opportunity to speak here. Thank you for the introduction, Richard. I'm going to speak to you about my project aimed at the characterisation of NKX2-5 mutations causative for congenital heart disease. So complications with heart development lead to a huge variety of congenital abnormalities which occur in approximately 40 babies per week in Australia alone, half of which have to undergo surgery. Underlying heart development is a complex web of temporally controlled gene expression, which is controlled and regulated by this handful of core transcription factors known as the cardiac gene regulatory network, which consists partly of NKX2-5, of GATA factors, TBX, HAND1 factors, as well as MEFs and SRF, and a few others. At the moment, there is absolutely no correlation between a dysfunction in these regulators and the disease phenotype which occurs. So, as I said, our research focuses on the role of NKX2-5 in this gene regulatory network and defining potentially mutations to this protein, to this transcription factor. So this resulted in a paper published in 2015 from our lab, which characterized two mutants of NKX. First of all, a point mutation within the homeodomain, or the DNA binding domain of NKX2-5, and a complete homeodomain deletion, which is a synthetic mutant, but a very severe mutant. And these both interestingly showed that they were still able to bind to a number of NKX wild-type targets, as shown by DAM-ID, as well as having a number of off-target effects, which was shown to be due to interrupted cofactor interactions. And these mutations also resulted in a disruption of motif binding grammar, or the DNA binding specificity of these mutants. So from here, we're now characterizing a further 12 mutants of NKX2-5, and we hypothesize that these mutants result in varied off-target effects, which are dependent on the residue or the domain affected. We also hypothesized that this off-target binding will result in a destabilization of the cardiac gene regulatory network, which may be due to either altered homo or heterodimerization, changed DNA binding affinity or specificity, or potentially via paracrine signaling effects. To test these hypotheses, we used DAM-ID, which is an alternative to CHIP for profiling protein-DNA interactions. The process uses bacterial methyltransferase fused to a DNA binding protein of interest. When your binding protein binds DNA, you get local methylation. We can then amplify these methylated fragments and subject them to either sequencing or microarray. So for the purposes of our work, we're using HL1 cardiomyocyte cell line to profile our NKX2-5 wild type and mutant binding using a proximal promoter microarray. So here are the mutants that we've characterized so far. These are 12 mutants, which are all, apart from one, which is a synthetic, associated with congenital heart disease and have been shown to be involved in the processes. So today I'm going to focus on a subset of these mutants, which are namely the mutations of DNA base contacting residues within the homeodomain. So the homeodomain of NKX2-5 interacts with DNA in a base to residue or backbone specific manner, primarily due to these DNA base contacting residues in the N-terminal tail of the homeodomain and within the third alpha helix of the homeodomain. So we're primarily focusing today on these DNA base contacting residues. So to reintroduce the nomenclature that we use, we consider the targets bound by wild type only as our A set, the mutual targets between both wild type and mutant as our B set, and the C set we see as the off targets, or the targets which are bound only by mutant. So if we convert these targets to a gene list and then associate this gene list with biological functions, or GO terms, we can plot a measure of functional similarity or functional disruption between our wild type on the x-axis and our mutant on the y-axis. And you can see that this gives us a surrogate measure of the functional similarity or disruption between mutant and wild type. So if you look here at our DNA base contacting mutations only, you see that they all have a large overlap with wild type, as can be shown in the B sets, but also they contain a large number of off targets, which are associated with varying degrees of functional disruption within these 3 mutants. So if we look in more depth at the individual target sets here, in this mutant specifically, we see that within the A set, these target genes are associated with primarily metabolic type functions. These are geoterms plotted in order of appearance. And you can see that they're primarily metabolic functions, suggesting that these mutants of DNA base contacting residues are potentially losing some degree of the periphery of this cardiac gene regulatory network. Then if we look at the targets in the B set and their associated gene ontology terms, you see that these are all associated with muscle or cardiac-related processes, suggesting that these mutants retain a degree of functionality, of cardiac-related functionality. Most interestingly, when we look at the C set targets of our DNA base contacting mutations, and this occurs in all of our mutants that are DNA base contacting, you see a gain of these cardiac-related functions shown by the geoterms here. And we define these as a gain of cardiac function, but this is not either an activation or a repression, but we define it as an increase in binding to cardiac-related targets. So if we try to go to explain this further, we look at motif discovery. So first of all, using de novo motif discovery, you see that the mutant here, the DNA base contact mutant, has a large number of off-target motifs which are not shared with the wild type. And then if we cluster these motifs based on their semantic similarity or the similarity of their position weight matrices, and then rank them in order, you see that in our C set we have absolutely no representation of the NKX motif, but we have a large representation of NF1, which is a novel cofactor of NKX2-5, and HAND1 motifs, which are highly represented here and largely represented within the de novo motif discovery. This data was corroborated by known motif enrichment. As you can see, this large unique C set of motifs within the C set only, and again we find HAND1 in this known motif enrichment. So focusing now on the role of HAND1 being one of the core transcription factors within the gene regulatory network, we see, using a protein-protein interaction assay or yeast-2 hybrid assay, that in fact this mutant N188K represents a super interactor with HAND1, suggesting that this mutant, like was shown in the 2015 paper, is being co-opted by HAND1 and drawn to off targets and therefore is potentially binding to HAND1 related targets or regulating them in some form, leading to this gain of cardiac binding function. Alternatively, these gain of function mutations of our DNA base contacting mutants may be due to an altered DNA binding specificity, as you can see with this protein binding microarray data here of the Y191C mutant. You see here is the standard NKX motif, and represented here. When this residue is mutated, you have a change in DNA binding specificity to the more archetypal homeobox type motif, which may again lead to our increase in gain of cardiac-related functions. And this validates the DAM-ID data, which was shown in 2015. And with that, I'd like to summarize that we've shown that these mutations of NKX2.5 lead to off-target effects, and that this may result in a destabilization of the cardiac gene regulatory network, either due to cofactor interaction, DNA binding specificity. These both may play roles in this. And we've shown that severity and functionality of these mutations may be associated to the function of the original residue which was mutated. So with that, I'd like to thank Professor Richard Harvey, Mirana Rami-Ellison, Romerick Bouveret and Ashley Wardenberg, Martha Bullock from Harvard Medical School, Stephanie Mohammed, and all of the Harvey Lab. And thank you all for staying with me to the end.